retrouvé leur adresse?
J'ai regardé dans ma liste de destinations. Tu penses pas que c'est là? Okay, hello gang. So I don't know if this is the beginning or the end of this 7-hour drive. Or I guess halfway through. Anyways, we're gonna spend 4 hours about going through this book. Why? Because it's a great book and there's no audio versions. We're looking at A Brief Tour of Human Consciousness by V.S. Ramachandran, author of Phantoms in the Brain, so phantom limb stuff. This book came out in 2004. I read it in 2005. Maybe 6, I don't know. Oh, first published in Great Britain in 2003. Copyrighted in 2004. So this is one of the first, if not the first, neuro book I ever read. And yeah, this is one of my favorite neuroscientists. And this is the book that Mom is starting with. But because there's no audio, here is the audio book. Okay, very good. So right now we're gonna read for an hour. And hopefully we will get through more than 40 pages. So then we can also read the notes, and we'll have the whole book done on this drive. Preface. My goal in writing this book has been to make neuroscience, the study of the brain, more accessible to a broad audience, to working men, as Thomas Huxley would have said. The overall strategy is to investigate neurological dysfunction caused by a change in a small part of a patient's brain and ask, why does this patient display these curious symptoms? What do the symptoms tell us about the workings of the normal brain? Can a careful study of these patients help us explain how the activity of 100 billion nerve cells in the brain gives rise to all the richness of our conscious experience? I've chosen to focus both on areas in which I have worked directly, such as phantom limbs, synesthesia, and visual processing, and ones that have a broad interdisciplinary appeal, in order, ultimately, to bridge the gap that now separates C.P. Snow's two cultures, the sciences and the humanities. The book emerged from the annual BBC Rafe lectures that I delivered in Great Britain in 2003. It was an honor for me to be invited to give these lectures, the first physician/experimental psychologist to do so since they were begun by Bertrand Russell in 1948. In the last 5 decades, these lectures have enjoyed a distinguished place in the intellectual and cultural life of the Western world. I was delighted to accept the invitation, knowing that I would be joining a long list of previous lecturers whose works inspired me as a teenager. I was delighted to accept the invitation, knowing that I would be joining a long list of previous lecturers. Sorry, are you thinking that during this 1 hour of reading that it's a good time to get a bag of chips and eat them? No? Oh, okay, my mistake. I was delighted to accept the invitation, knowing that I would be joining a long list of previous lecturers whose works inspired me as a teenager. Peter Medawar, Arnold Toynbee, J. Robert Oppenheimer, John Galbraith, and Russell, to mention only a few. Okay, sorry for the interruption. Decided to email Ramachandran, wanted to do it for a long time. So wrote a little email to him. We'll send that off on Monday. And to Patricia Churchland. Okay, back into it. Okay, so we're in the preface. Okay. And we're in paragraph 2. I was delighted to accept the invitation, knowing that I would be joining a long list of previous lecturers whose works inspired me as a teenager. Peter Medawar, Arnold Toynbee, J. Robert Oppenheimer, John Galbraith, and Russell, to mention only a few. Oh, and Russell. We talked about Bertrand Russell. To mention only a few. I realized that theirs would be a tough act to follow, given their towering stature and the pivotal role that many of them played in defining the intellectual ethos of our age. Even more daunting was the requirement that I would have to make the lectures not only interesting to the specialist, but also intelligible to the common people, thereby fulfilling Lord Wraith's original mission for the BBC. Given the enormous amount of research on the brain, the best I could do was to provide an impressionistic survey rather than try to be comprehensive. In doing this, I was worried I might have oversimplified many of the issues involved, and so run the risk of annoying some of my specialist colleagues. But as Lord Wraith himself once said, there are some people whom it is one's duty to annoy. Chapter 3, based on the third lecture, deals with an especially controversial subject, the neurology of artistic experience, neuroaesthetics, that is usually considered off limits by scientists. I take a stab at it just for fun and to indicate how a neuroscientist might approach this problem. I make no apology for the fact that it is speculative. As Peter Medawar said, all good science begins as an imaginative excursion into what might be true. That's really nice. Speculation is fine, provided it leads to testable predictions, and so long as the author makes it clear when he is merely speculating, skating on thin ice, as opposed to when he's on solid ground. I have taken pains to preserve this distinction throughout the book, often adding qualifying remarks in what have become extensive endnotes. There is also a tension in the field of neurology between the single case study approach, the intensive study of just 1 or 2 patients with a syndrome, and sifting through a large number of patients and doing a statistical analysis. The criticism is sometimes made that it is easy to be misled by single strange cases, but this is nonsense. Most of the syndromes in neurology that have stood the test of time, for example, the major aphasias, language disturbances, amnesia, explored by Brenda Milner, Elizabeth Warrington, Larry Squire, and Larry Weiskrantz, cortical color blindness, neglect, blind sight, split brain syndrome, commissurotomy, etc., were initially discovered by a careful study of single cases. And I don't know of even one that was discovered by averaging results from a large sample. The best strategy, in fact, is to begin by studying individual cases and then to make sure that the observations are reliably repeatable in other patients. This is true for a majority of findings described here, such as phantom limbs, the Capgras or imposter delusion, synesthesia, and neglect. The findings are remarkably consistent across patients and have been confirmed in several laboratories. Many colleagues and students often ask me when I became interested in the brain and why. It's not easy to trace the lineage of one's interest, but I'll give it a shot. I have been interested in science from about the age of 11. I remember being a somewhat lonely child and socially awkward, although I did have one very good science playmate in Bangkok. Samtao Cookie Suchareko. I always felt companioned by nature, the journal, and perhaps science was my retreat from the social world with all its arbitrariness and mind-numbing conventions. I spent a lot of time collecting seashells and geological specimens and fossils. I enjoyed dabbling in ancient archaeology, cryptology, the Indus script, comparative anatomy, and paleontology. I found it endlessly fascinating that the tiny bones inside your ears, which we mammals use for amplifying sounds, had originally evolved from the jawbones of reptiles. As a schoolboy, I was passionate about chemistry and often mixed chemicals just to see what would happen. A burning piece of magnesium ribbon could be plunged into water. It would continue to burn underwater by extracting the oxygen from H2O. Tons of fun. Another passion was biology. I once tried placing various sugars, fatty acids, and individual amino acids inside the mouths of Venus flytraps to see what triggered them to stay shut and secrete digestive enzymes. And I did an experiment to see if ants would hoard and consume saccharin, showing the same fondness for it that they do as they do for sugar. Would the saccharin molecule fool their taste buds the same way it fools ours? All these pursuits, Victorian in inspiration, are quite remote from the neurology and psychophysics I specialize in now. Yet, these childhood preoccupations must have left a mark that profoundly influences my adult personality and my style of doing science. While engaged in such pursuits, I felt that I was in my own private playground, my own parallel universe inhabited by Darwin and Cuvier and Huxley and Owen and William Jones and Champollion. For me, these people were more alive than most real people I knew. Perhaps this escape into my own private world made me feel special rather than isolated, weird, or different. It allowed me to rise above the tedium and monotony, the humdrum existence that most people call a normal life. To a place where, to quote Russell, one, at least, of our nobler impulses can escape from the dreary exile of that actual world. Such an escape is especially encouraged at the University of California, San Diego, an institution that is both venerable and vibrantly modern. Its neuroscience program was recently ranked #1 in the country by the National Academy of Sciences. If you include the Salk Institute and Gary Edelman's Neurosciences Institute, there is a higher concentration of eminent neuroscientists in La Jolla's Neuron Valley than anywhere else in the world. Okay. If you include the Salk Institute and Jerry Edelman's Neurosciences Institute, there is a higher concentration of eminent neuroscientists in La Jolla's Neuron Valley than anywhere else in the world. Whoa. I can't think of a more stimulating environment for anyone interested in the brain. Whoa. Science is most fun when it is still in its infancy, because its practitioners are still driven by curiosity and it hasn't become professionalized into just another 9-to-5 job. Unfortunately, this is no longer true for many of the most successful areas of science, such as particle physics or molecular biology. It is now commonplace to see a paper in Science or Nature co-written by 30 authors. For me, this takes some of the joy out of it, and I imagine it does for the authors too. This is one of the two reasons I instinctively gravitate toward old-fashioned Geschwindian neurology, where it is still possible to ask naive questions starting from first principles. The kinds of very simple questions that a schoolboy might ask, but are embarrassingly hard for experts to answer. The kinds of very simple questions that a schoolboy might ask, but are embarrassingly hard for experts to answer. It's a field where it's still possible to do Faraday-style research and come up with surprising answers. Indeed, many of my colleagues and I see it as an opportunity to revive the golden age of neurology, the age of Charcot, Hewling-Jackson, Henry Head, Luria, and Goldstein. The second reason I chose neurology is more obvious. It's the same reason you picked up this book. As human beings, we are more curious about ourselves than about anything else. And this is a research enterprise that takes you right into the heart of the problem of who we are. I got hooked on neurology after examining my very first patient in medical school. He was a man with a pseudobulbar palsy, a kind of stroke, who alternately laughed and wept uncontrollably every few seconds. It struck me as an instant replay of the human condition. Were these just mirthless joy and crocodile tears, I wondered? Or was he actually feeling alternately happy and sad, the same way a manic depressive might, but on a compressed time scale? We will be considering many such questions throughout the book. What causes phantom limbs? How do we construct a body image? Are there artistic universals? What is a metaphor? Why do some people see musical notes as colored? What is hysteria? Some of these questions I will answer, but the answers to the others remain tantalizingly elusive, such as the big question, what is consciousness? But whether I answer them or not, this book at least whets your appetite to learn more about this fascinating field. It will have served its purpose. Endnotes, a glossary, a bibliography, and an index are provided at the end for the benefit of those who wish to probe these topics more deeply. As my colleague Oliver Sacks said of one of his books, "The real book is in the endnotes, Rama." I would like to dedicate this work to the patients who volunteered to endure hours of testing at our center. I have often learned more from my conversations with them, despite their damaged brains, than from my learned colleagues. I guess we'll probably take a break between each chapter, so take a break.
Et bah parfait, on a pu faire notre truc.
Hello. Chapter 1, A Pain in the Brain. The history of mankind in the last 300 years has been punctuated by major upheavals in human thought that we call scientific revolutions. Upheavals that have profoundly affected the way in which we view ourselves and our place in the cosmos. First, there was the Copernican revolution. The notion that far from being the center of the universe, our planet is a mere speck of dust revolving around the sun. Then there was the Darwinian revolution, culminating in the view that we are not angels, but merely hairless apes, as Thomas Henry Huxley once pointed out. And third, there was Freud's discovery of the unconscious. There is Freud's discovery of the unconscious, the idea that even though we claim to be in charge of our destinies, most of our behavior is governed by a cauldron of motives and emotions of which we are barely conscious. Your conscious life, in short, is nothing but an elaborate post hoc rationalization of things you really do for other reasons. Gosh, that's like, I don't want to make a lot of comments, but that is like the most important page you could basically ever read, except it doesn't mention cognitive errors. But gosh, it just really puts it in perspective. And this is something that I don't know how much I've been saying in videos, but it's something that's been screaming in my head lately. Is this timeline of, you know, putting in perspective that the stuff I'm working on this channel, the stuff my group's working on, the stuff that you and I are working on, has been sort of in the making the last few hundred years, and is only now able to hit sort of its final steps. What are those final steps? Well, us putting all this stuff together and making the transition away from our intuitive sense of self to a more informed and useful sense of self. We went from, you know, before 300 years ago, thinking that we were these angels, right, put here by God, that we were the whole universe was for us, that we knew everything that we were thinking and doing and so on. So first, Copernicus comes along, goes, hey, we're not the center of the universe. We're not even the center of any local system that we could name. Then, wait a minute, we're not angels. So not only was this universe not built for us, we are also like biological things. And then, actually, we're not even automatically self-knowing biological things. Or we're not angels, and we're biological things, but also we don't know like what we're thinking. We're not necessarily thinking correctly. Well, I mean, that's like the fourth thing. First of all, there's a whole bunch of processes that we don't know. So that's Freud. And then boom, fourth step, Kahneman saying, oh, yo, I mean, lots of people have talked about mental errors, but Kahneman giving it to us real good. Being like, hey, you know all that unconscious stuff going on? That's where the decisions are happening, and it's according to these shortcuts, these heuristics, not this rule this really sort of reasoned analysis that you think has been happening. So now it's like now that we can understand that. And also we've only had the technology recently to really be able to confirm that, you know, there's just no more room for doubt that this talking part of me is a brain, that this thinking, that my entire experience is neural. Yeah, of course, what's cool is that most people accept what Copernicus said. And then maybe almost half of people accept what Darwin said. Unfortunately, the majority of people have not yet accepted what Freud told us about all these unconscious things. Maybe. And then even fewer have accepted that these unconscious things could actually be wrong, right? All kinds of people, they talk about their unconscious, their intuition and stuff, but they think it's this magical truth thing. All right, anyway. Okay, no more. I'm gonna beep. We really don't. It'll take 25 hours to read this book okay, back into it. Okay, page 2. But now we are poised for the greatest revolution of all. And that last line. Okay, so anyway, so he did the Copernicus, he did Darwin, he did Freud. And then he says, your conscious life, in short, is nothing but an elaborate post hoc rationalization of things you really do for other reasons, and not necessarily good ones. But now we are poised for the greatest revolution of all. understanding the human brain. This will surely be a turning point in the history of the human species, for unlike those earlier revolutions in science, this one is not about the outside world, not about cosmology or biology or physics, but about ourselves, about the very organ that made those earlier revolutions possible. And I want to emphasize that these insights into the human brain will have a profound impact, not just on scientists, but also on the humanities. And indeed, they may even help us bridge what C.P. Snow called the two cultures, science on the one hand, and arts and philosophy and humanities on the other. Given the enormous amount of research on the brain, all I can do here is to provide a very impressionistic survey rather than try to be comprehensive. The five chapters cover a very wide spectrum of topics, but two recurring themes run through all of them. The first broad theme is that by studying neurological syndromes, which have been largely ignored as curiosities or mere anomalies, we can sometimes The first broad theme is that by studying neurological syndromes, which have been largely ignored as curiosities or mere anomalies, we can sometimes acquire novel insights into the functions of the normal brain, how the normal brain works. The second theme is that many of the functions of the brain are best understood from an evolutionary vantage point. The human brain, it has been said, is the most complexly organized structure in the universe. And to appreciate this, you just have to look at some numbers. The brain is made up of 100 billion nerve cells, or neurons, which form the basic structural and functional units of the nervous system. Each neuron makes something like 1,000 to 10,000 contacts with other neurons, and these points of contact are called synapses. It is here that exchange of information occurs. Based on this information, it has been calculated that the number of possible permutations and computations of brain activity, in other words, the numbers of brain states, exceeds the number of elementary particles in the known universe. Even though it is common knowledge, it never ceases to amaze me that all the richness of our mental life, all our feelings, our emotions, our thoughts, our ambitions, our love lives, our religious sentiments, and even what each of us regards as his or her own intimate, private self, is simply the activity of these little specks of jelly in our heads, in our brains. There is nothing else. Given this staggering complexity, where does one even begin? Well, let's start with some basic anatomy. In the 21st century, most people have a rough idea of what the brain looks like. It has two mirror image halves called the cerebral hemispheres and resembles a walnut sitting on top of a stalk called the brainstem. I think of it as a boxing glove. And you can think of a little skinny little wrist coming out. Each hemisphere is divided into four lobes: the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. The occipital lobe in the back is concerned with vision. Damage to it can result in blindness. The temporal lobe is concerned with hearing, emotions, and certain aspects of visual perception. The parietal lobes of the brain at the sides of the head are concerned with creating a three-dimensional representation of the spatial layout of the external world and also of your own body within that three-dimensional representation. And lastly, the frontal lobes, perhaps the most mysterious of all. They are concerned with some very enigmatic aspects of the human mind and human behaviors, such as your moral sense, your wisdom, your ambition, and other activities of the mind which we know very little about. More like that. There are several ways of studying the brain, but my approach is to look at people who have had some sort of damage or change to a small part of their brain. Interestingly, people who have had a small lesion in a specific part of the brain do not suffer an across-the-board reduction in all their cognitive capacities, no blunting of their mind. Instead, there's often a highly selective loss of one specific function while other functions are preserved intact, which is a good indication that the affected part of the brain is somehow involved in mediating the impaired function. I could cite many examples, but here are some of my favorites. First, I've always pronounced it prosopagnosia, but I see it's an O. Prosopagnosia, or face blindness. When a structure called the fusiform gyrus in the temporal lobes is damaged on both sides of the brain, something like that. When a structure called the fusiform gyrus in the temporal lobes is damaged on both sides of the brain, the patient can no longer recognize people's faces. The patient can still read a book, so is not blind, and is not psychotic or mentally disturbed in any way, but is simply no longer able to recognize people by just looking at the face. Prosopagnosia is very well known, but there is another syndrome that is quite rare, the Capgras syndrome. A patient I saw not long ago had been in a car accident, sustaining a head injury, and was in a coma. He came out of the coma after a couple of weeks and was quite intact neurologically when I examined him, but he had one profound delusion. He would look at his mother and say, "Doctor, this woman looks exactly like my mother, but she isn't. She is an imposter." Why would this happen? Bear in mind that this patient, who I will call David, is completely intact in other respects. He is intelligent, alert, fluent in conversation, and not emotionally disturbed in any other way. The standard explanation for the Capgras delusion, found in older psychiatry textbooks, is a Freudian one. Okay, resuming. The standard explanation for the Capgras delusion, found in older psychiatry textbooks, is a Freudian one. According to this view, during infancy, all of us men have a strong sexual attraction toward our mothers, the so-called Oedipus complex. But as we grow up, the cortex becomes more highly developed and inhibits or represses these latent urges originating in the limbic emotional core of the brain. Thank God for that, otherwise we would all be permanently sexually aroused by our mothers. But then along comes a blow to the head, and these repressed sexual urges come flaming to the surface, so that suddenly the patient finds himself sexually aroused by his mother. The only way he can rationalize these forbidden feelings is to say, if it's my mom, why am I sexually turned on? It must be an imposter. Hence the delusion. This is an ingenious explanation, as indeed all Freudian explanations are, but it doesn't work. I have seen a patient having the same delusion not only about his mother, but also about his pet poodle. "This isn't Fifi, doctor. It's some other dog that looks like Fifi." Clearly, the Freudian explanation cannot explain this without invoking something absurd like the latent bestiality of all humans, which would be too far-fetched even by the notoriously lax intellectual standards of Freudian psychology. So what really causes Capgras syndrome? To understand this disorder, you have to first realize that vision is not a simple process. When you open your eyes in the morning, it's all out there in front of you, and so it's easy to assume that vision is effortless and instantaneous. But in fact, within each eyeball, all you have is a tiny, distorted, upside-down image of the world. This excites the photoreceptors in the retina, and the messages then go through the optic nerve to the back of your brain, where they're analyzed in 30 different visual areas. Only after that do you begin to finally identify what you're looking at. Is it your mother? Is it a snake? Is it a pig? And that process of identification takes place partly in a small brain region called the fusiform gyrus, the region which is damaged in patients with face blindness or prosopagnosia. Finally, once the image is recognized, the message is relayed to a structure called the amygdala, sometimes called the gateway to the limbic system, the emotional core of your brain, which allows you to gauge the emotional significance of what you are looking at. Is this a predator? Is it a prey which I can chase? Is it a potential mate? Is it my mother? Or is it my departmental chairman I have to worry about? A stranger who is not important to me. That I have to worry about. A stranger who is not important to me, or something utterly trivial like a piece of driftwood. What is it? In David's case, perhaps the fusiform gyrus and all the visual areas are completely normal. So his brain cells tell him that the woman he sees looks like his mother. But to put it crudely, okay, I'm going to do it once and one time only. I, of course, object to any phrasing that involves the brain telling the person something, right? The more accurate phrasing would be that the brain knows a thing, or this part tells another part, or this part knows it, and tells another part, something like that. But his brain tells him, I guess I won't say it's meaningless, but I object because of what it can mean to people. Anyway, so to put it crudely, the wire that goes from the visual centers to the amygdala, to the emotional centers, is cut by the accident. So he looks at his mother and thinks, she looks just like my mother. But if it's my mother, why don't I feel anything toward her? No, this can't possibly be my mother. It's some stranger pretending to be my mother. This is the only interpretation that makes sense to David's brain, given the peculiar disconnection. How can an outlandish idea like this be tested? My student Bill Hurston and I in La Jolla, and Hayden Ellis and Andrew Young in England, did some very simple experiments measuring galvanic skin response. See Chapter 5. We found, sure enough, that in David's brain there was a disconnection between vision and emotion as predicted by our theory. Between vision and emotion as predicted by our theory. Even more amazing is that when David's mother phones him, he instantly recognizes her from her voice. There is no delusion. Yet, if an hour later his mother were to walk into the room, he would tell her that she looked just like his mother but was an imposter. The reason for this anomaly is that a separate pathway leads from the auditory cortex in the superior temporal gyrus to the amygdala, and that pathway perhaps was not cut by the accident. So auditory cognizance remains intact while visual cognizance has disappeared. This is a lovely example of the sort of thing we do. A cognitive neuroscientist in action. Of how you can take a bizarre, seemingly incomprehensible neurobiological syndrome, a patient claiming that his mother is an imposter, and then come up with a simple explanation in terms of the known neural pathways in the brain. Our emotional response to visual images is obviously vital to our survival, but the existence of connections between visual brain centers and limbic systems the limbic system or emotional core of the brain also raises another interesting question. What is art? How does the brain respond to beauty? Given that these connections are between vision and emotion, and that art involves an aesthetic emotional response to visual images, surely these connections must be involved. And this forms the subject of a later chapter. Are these intricate connections in the brain laid down by the genome in the fetus, or are they acquired in early infancy as we begin to interact with the world? This is the so-called nature-nurture debate and is central to my next example, phantom limbs. Most people know what is meant by a phantom limb. A patient has an arm amputated because it has a malignant tumor or has been irreparably damaged in an accident, but the patient continues to feel the presence of the amputated arm. A famous example concerns Lord Nelson, who vividly felt a phantom arm long after his real one had been lost in battle. He actually used it in a somewhat flawed argument for the existence of a non-corporeal soul. For if an arm can survive physical annihilation, he asked, why not the whole body? I once asked a patient whose arm had been amputated above the left elbow. No, I once had a patient whose arm had been amputated above the left elbow. Boop. So this is what the patient had, as you see in the image. He sat in my office blindfolded while I gently touched different areas of his body and asked him to say where I was touching him. All went as expected until I touched his left cheek, at which point he exclaimed, "Oh my God, you're touching my left thumb." His missing phantom thumb, in other words. He seemed as surprised as I was. Touching his upper lip produced sensation in his phantom index finger. So this is the left arm that was cut, right? And touching his lower jaw provoked sensations in his phantom little finger. There was a complete systematic map of the missing phantom hand draped on his face. Why does this happen? Like the Capgras delusion, phantom limbs are a mystery that would have intrigued Sherlock Holmes. What on earth is going on here? The answer lies, again, in the anatomy of the brain. The touch signals from the entire skin surface on the left side of the body are mapped onto the right cerebral hemisphere on a vertical strip of cortical tissue called the post-central gyrus. Actually, there are several maps, but for ease of representation, we can assume there is only one map, called S1, on the post-central gyrus. Wait, S1 or SI? Because this is the primary somatosensory, so it's S1, right? That's what that says. What would the I represent? Called S1 on the post-central gyrus. This is a faithful representation of the entire body surface. Oh, right. Okay. Oh, yeah. This is a faithful representation of the entire body surface, almost as if I gotta see if I'm still recording. Okay. This is a faithful representation of the entire body surface, almost as if there were a little person draped on the surface of the brain. Figure 1.5. We call this the Penfield homunculus, and for the most part, it is continuous, which, after all, is what one means by a map. But there is one peculiarity. The representation of the face on this map on the surface of the brain is right next to the representation of the hand, not near the neck in its expected position. The head is dislocated. Why this is so is unclear. Perhaps it is something to do with the phylogeny, or the way in which the brain develops in early fetal life, or in early infancy, but dislocated it is. This gave me the clue to what was happening. When an arm is amputated, no signals, boop, okay, no more signals from here are going to here, right? When an arm is amputated, no signals are received by the part of the, oh, well, it should be this side, right? By the part of the brain's cortex. When an arm is amputated, no signals are received by the part of the brain's cortex corresponding to the hand. It becomes hungry for sensory input, and the sensory input from the facial skin now invades the adjacent vacated territory corresponding to the missing hand. Signals from the face are then misinterpreted by higher centers in the brain as arising from the missing hand. The specificity of these signals is so compelling that an ice cube or warm water applied to the face will produce a cold or warm phantom digit. One patient, Victor, when the water began to trickle down his face, also felt it trickling down his phantom arm. When he raised his arm, he was amazed to feel the trickle going up his phantom arm. Or, yeah, he went like this. Whoa. Well, I don't know. Did he have no arm? In which case he just like moved his shoulder? I don't know. But anyway, when he did what he'd have to do to raise his arm, he was amazed to feel the trickle going up his phantom, contrary to the laws of physics. To test our remapping or cross-wiring hypothesis directly, we used the brain imaging technique called MEG, or magnetoencephalography. This shows which parts of the brain are stimulated when various parts of the body are touched. Wow, because that's clear evidence as anything. I totally forgot it. I don't think about phantom limbs enough. You're not your whole body, bro. Obviously, you're your brain. I just tweak your brain a little bit. Well, a little bit. And all of a sudden when I touch your nose, you think I'm touching a hand that you don't even have. Ugh. Now, there's a reason God put me in the past. So this shows which parts of the brain are stimulated when various parts of the body are touched. Sure enough, we found that in Victor and other arm amputees like him, touching his face activated not only the face area in the brain, but also the hand region of the Penfield map. All right, no, I should be reacting. I was reacting as a scientist. And then we see activity in both areas. Whoa. This is very different from what is seen in a normal brain, where touching the face activates only the facial region of the cortex. There's obviously been some cross-wiring in Victor's brain, and this is important because it permits us to correlate the changes in brain anatomy, changes in brain sensory maps, with the phenomenology. This link between physiology and psychology is one of the major goals of cognitive neuroscience. The discovery also has broader implications. One of the things all medical students learn is that connections in the brain are laid down in the fetus or in early infancy, and that once they are laid down, there's nothing much that can be done to change these connections in an adult. That's why when there's damage to the nervous system, such as is caused by a stroke, there is so little recovery of function. It is also why neurological ailments are notoriously difficult to treat, or at least that's what we were taught. What I have seen flatly contradicts this view and suggests that there is a tremendous amount of plasticity or malleability even in the adult brain, and this can be demonstrated in a 5-minute experiment on a patient with a phantom limb. It isn't yet clear how this plasticity of body maps can be harnessed in the clinic, but I'll mention another example to show how some of these ideas can be clinically useful. Some patients can move their phantom limbs and will say, "Oh, it's waving goodbye," or "It's shaking hands with you." But in many other patients, the phantom arm feels paralyzed, frozen stiff, in cement, or won't budge an inch. Often the phantom hand goes into painful, involuntary clenching spasms, or is fixed in an awkward, painful position which the patient is unable to change. We have discovered that some of these patients had pre-existing nerve damage before the amputation. For example, the arm had been paralyzed and lying in a sling. After amputation, the patient is stuck with a paralyzed phantom, as if the paralysis is carried over into the phantom. Perhaps when the arm was intact but paralyzed, every time the front of the brain sent a command to the arm saying move, it was getting visual feedback saying, no, it won't move. Somehow, this feedback becomes imprinted on the circuitry in the parietal lobe or somewhere else in the brain. We call this learned paralysis. How could this highly speculative idea be tested? Perhaps if a patient were given visual feedback that the phantom was obeying the brain's commands, the learned paralysis could be unlearned. We propped up a mirror vertically on a table in front of a prone patient, and Sarah said, no, you're not allowed to prop up mirrors on a table. Sarah? Huh? Oh, did you miss that? Oh, it's just a really funny moment. It's all right. No, I'll tell you later. We propped up a mirror vertically on a table in front of a prone patient so that it was at right angles to his chest, and asked him to position his paralyzed phantom left arm on the left of the mirror and mimic its posture with his right hand, which was on the right side of the mirror. We then asked him to look into the right-hand side of the mirror so that he saw the mirror reflection of his intact hand optically superimposed on the felt location of the phantom, figure 1.7. We then asked him to try to make symmetrical movements of both hands, such as clapping or conducting an orchestra, while looking in the mirror. Imagine his amazement, and ours, when suddenly he not only saw the phantom move, but felt it move as well. I've repeated this experiment with several patients, and it seems that the visual feedback animates the phantom so that it begins to move as never before, often for the first time in years. Many patients have found that this sudden sense of voluntary control and movement in the phantom produces relief from the spasm or awkward position or awkward posture that was causing much of the agonizing pain in the phantom. Relief from a phantom pain using a mirror is surprisingly enough, but can the same trick be applied to real pain in an intact arm or leg? Even though we usually think of pain as one thing, there are at least two different types which may have evolved from different functions. Acute pain evolved to allow reflexive withdrawal from, for example, fire, and probably also to teach avoidance of harmful pain-producing objects such as thorns. Chronic pain, as in a fracture or a gangrene, is a different thing altogether. It evolved to reflexively immobilize the arm, so letting it rest and remain out of harm's way until it's fully healed. Ordinarily, pain is a very useful adaptive mechanism, a gift, not a curse. But sometimes the mechanism backfires. We often see patients with a condition called chronic regional pain type 1, which includes the bizarre clinical syndrome of reflex sympathetic dystrophy, or RSD. In RSD patients, reflex sympathetic dystrophy, in RSD patients, what begins as a minor injury, a bruise or a little insect sting or fracture of a fingertip, leads to the entire arm becoming excruciatingly painful, completely immobilized, inflamed, and swollen, grossly out of proportion to the inciting event, and it lasts forever. The evolutionary framework helps us to understand how this might come about. Remember, the original purpose of chronic pain is a temporary immobilization to allow recovery. So when the brain sends a motor command to the arm, there is intense pain preventing further movement. This is ordinarily adaptive, but I suggest that it sometimes malfunctions and leads to what I call learned pain. The very act of attempting to move the arm, the motor command signal itself, becomes pathologically associated with excruciating pain. As a result, even after the inciting event has long disappeared, the patient still has a pseudo-paralysis caused by learned pain. In 1995, I suggested that this type of pathological chronic pain may also benefit from mirror visual feedback. Imagine the patient sees the reflection of a normal hand superimposed optically on the painfully immobilized abnormal one. If the normal hand is now moved, while partially attempting to move the painful one, the patient will see the bad arm suddenly springing to life and moving quite freely. Perhaps this would help RSD patients to unlearn the spurious connection in their brains between arm movement and pain, thereby eliminating the pain and returning mobility. In 1995, this was no more than a far-fetched idea. But recently, McCabe et al., 2003, tried the mirror procedure on 9 patients in placebo-controlled clinical trials. The pain went away completely and mobility returned in many of the patients who used mirrors, whereas the control group, who used plexiglass, experienced no benefit at all. This result is so surprising that I would have been skeptical had not Patrick Wall, arguably the world's leading expert on both pain and placebos, been one of the authors. If confirmed, this result promises a new and effective treatment for at least some patients with chronic pain. The cross-wiring in the brain that sometimes results from amputation can also occur owing to a gene mutation. Instead of the brain modules remaining segregated, they become accidentally cross-wired, resulting in a curious condition called synesthesia, first clearly documented by Francis Galton in the 19th century. Synesthesia, which appears to be genetically transmitted, results in a mingling of the senses. For example, hearing a particular musical note might invoke a particular color. C sharp is red, F sharp is blue, etc. Visually perceived numbers can produce a similar effect. 5 might always be seen as red, 6 always green, 7 always indigo, 8 always yellow. Synesthesia is surprisingly common, affecting 1 in 200 people. What causes this mixing of signals? All right, I'm sure he's going to say it, but I do have to say it. You're not a synesthete. Okay, maybe you are. But lots of people do associate colors with certain seasons, days, numbers, letters. I know I do. But that's not what synesthesia is. Synesthesia is when it's constant and when you're like really experiencing it. So like the number 5 is kind of yellow for me. I do think it's kind of always been yellow, but that's probably more likely like a fridge magnet thing. I'm not a synesthete, okay? Well, I am pretty good at metaphor and so on, but no, like when I see the word number 5, it's not just when you think of it. It's like you see it written on the page and it frickin' looks yellow to you. I think that's accurate. Okay, so anyway, it's 0.5%. So what causes this mixing of signals? A student of mine, Ed Hubbard, and I were looking at brain atlases, specifically at the fusiform gyrus, where color information is analyzed, and also faces. We saw that the number area of the brain, which represents visual graphemes of numbers, is also in the fusiform gyrus, almost touching the number area. It seems likely that, just as amputation can produce cross-wiring between the face and the hand, synesthesia is caused by cross-wiring between the number and color areas in the fusiform gyrus due to an inherited genetic abnormality. I just had to make sure that we were talking about the same fusiform gyrus. Because obviously the fusiform gyrus, so a gyrus is a, the gyrus is, it's just basically a raised surface. So, you know, the brain is like this, right? Hills and valleys. So the hills are gyri and the valleys are sulci, okay? I can't believe I have to check this. Correct, yeah. So the fusiform gyrus, this little region, I'd say probably about that big, bro. Maybe, yeah, I don't know. And, you know, there's a number area, color area, a face area, and so on, okay? So then you have the neurons a little too mixed up between the color area and the face area. In fact, I don't know why they would ever really need to communicate. And you've got this synesthesia, perhaps. Even though synesthesia was described by Galton 100 years ago, the phenomenon never made it into mainstream neuroscience. It was often assumed that these people were either just crazy or simply trying to draw attention to themselves. Or maybe it had something to do with childhood memory, refrigerator magnets, or a learning book in which 5 was red, 6 was blue, 7 was green, ... But if that were the case, how could it run in families? My colleagues and I wanted to show that synesthesia is a real sensory phenomenon, not mere imagination or memory. We devised a simple computer display, a number of black 5s scattered on a white background. Embedded among those 5s were a number of 2s forming a hidden shape, figure 1.8. Since these are computer generated, the 2s are just mirror images of the 5s. Most people looking at this pattern see only a random jumble of numbers, but a synesthete sees the 5s as green and the 2s as forming a red shape. It's forming a red shape conspicuously visible against a forest of green, shown schematically in figure 4.1. The fact that synesthetes can more easily identify these shapes than normal people suggests that they are not crazy, but experiencing a genuine sensory phenomenon. It also rules out memory association or some high-level cognitive phenomenon. Our group in La Jolla, as well as Jeffrey Gray and Mike Morgan and others in London, have conducted experiments to test the idea that there is cross-wiring in the brain. We have shown that there is activation of the fusiform gyrus in the color area when these people are shown numbers in black and white. In normal people, the color area is activated only if they are shown colored numbers. Phantom limb, synesthesia, and Capgras syndrome can at least be partly explained in terms of neural circuitry. But I once encountered someone with an even more bizarre syndrome called pain asymbolia. To my amazement, this patient responded to a pain stimulus not with an ouch, but with ha-ha laughter. Here's the ultimate irony: a human being laughing in the face of pain. Why would anyone do this? First, we need to answer an even more basic question: why does anybody laugh? Clearly, laughter is hardwired. It's a trait in all humans. Every society, every civilization, every culture has some form of laughter and humor, except for Germans. But why did laughter evolve through natural selection? What biological purpose does it serve? The common denominator of all jokes is a path of expectation that is diverted by an unexpected twist, necessitating a complete reinterpretation of all the previous facts. The punchline. Obviously, a sudden twist per se is not sufficient for laughter. Otherwise, every great scientific discovery that generates a paradigm shift would be greeted with hilarity, even by those whose theory had just been disproved. No scientist would be amused if you disproved his theory. Believe me, I've tried. Reinterpretation alone is insufficient. The new model must be inconsequential. The newly interpreted thing, what's replacing the old model. It must be inconsequential. For example, a portly gentleman walking toward his car slips on a banana peel and falls. If he breaks his head and blood spills out, obviously you're not going to laugh. You are going to rush to the telephone and call an ambulance. But if he simply wipes off the goo from his face, looks around him, and then gets up, you start laughing. Okay, yeah. Or you might start laughing immediately, but that's because you think it's not a big fall, right? And then as soon as you start seeing blood, you go, oh, fuck, not funny. The reason is, I suggest, because now you know it's inconsequential. No real harm has been done. I would argue that laughter is nature's way of signaling that it's a false alarm. Why is this useful from an evolutionary standpoint? I suggest that the rhythmic staccato sound of laughter evolved to inform our kin who share our genes. Don't waste your precious resources on this situation. It's a false alarm. Laughter is nature's okay signal. But what does this have to do with my pain asymbolia patient? Let me explain. When we examined his brain using a CT scan, we found there was damage close to the region called the insular cortex on the sides of the brain. The insular cortex receives pain signals from the viscera, and from the skin. That's where the raw sensation of pain is experienced. False. What do you mean? The insular cortex. I thought you were talking about the skin. I see. Okay. The insular cortex receives pain signals from the viscera and from the skin. And it's where the raw sensation of pain is experienced. But there are many layers to pain. It is not just a unitary thing. From the insular cortex, the message goes to the amygdala, encountered earlier in the context of the Capgras syndrome, and then to the rest of the limbic system, and especially the anterior cingulate, where we respond emotionally to the pain. We experience the agony of pain and take the appropriate action. So perhaps in this patient, the insular cortex was normal, so he could feel the pain, but the wire that goes from the insula to the rest of the limbic system in the anterior cingulate was cut, a disconnection similar to that seen in the Capgras patient. Such a situation would produce the two key ingredients required for laughter and humor. One part of the brain signals a potential danger, but the very next instant another part, the anterior cingulate, does not receive a confirmatory signal, thereby leading to the conclusion, oh, it's a false alarm. Hence, the patient starts laughing and giggling uncontrollably. The same sort of thing happens in tickling, which may be a sort of crude playtime rehearsal for adult humor. An adult approaches a child with his hands extended menacingly toward vulnerable parts of the child's body, but then anticlimactically deflates the potential threat with gentle stimulation and a coochie. I can't say coochie as I'm picturing tickling a child, bro. Especially you've talked about rehearsal for adult humor. I don't know what's going on. Well, anyway. This takes the same form as mature adult humor, a potential threat followed by a deflation. Okay, when I see adult, I think like adult. Okay, I got you. Anyway, so the examples we have considered so far suggest that syndromes such as phantom limb, the Capgras delusion, or pain asymbolia can help us understand the neural basis, functional logic, and evolution of many otherwise mysterious aspects of our minds. This is true whether you are considering little quirks of human behavior, such as our response to tickling, or the loftiest question of all, the neural basis of the self. There is something distinctly odd about a hairless, neotenous primate that has evolved into a species that can look back over its shoulder to ponder its origins. Odder still, the brain can not only discover how other brains work, but also ask questions about itself. Who am I? What is the meaning of my existence? Why do I laugh? Why do I dream? Why do I enjoy art, music, and poetry? Oh my gosh. Does my mind consist entirely of the activity of neurons in me? Well, he said my brain, but I couldn't do it. Because it was just doing a perfect thing of the brain asking questions about itself, and then one of the questions is about my brain. Come on. So I'm gonna paraphrase to me. Does my mind do I consist entirely of the cellular activities of me? No. Do I consist entirely of the activity of my neurons? If so, what scope is there for free will? It is the peculiar recursive quality of these questions, as the brain struggles to understand itself, that makes neurology so fascinating. Oh yeah, by the way, I should just finish this paragraph, or this chapter. I was just going to say, I think he's MD PhD, by the way, but we'll check that in a second. The prospect of answering these questions in the millennium is both exhilarating and disquieting, but it's surely the greatest adventure that our species has ever fucking embarked on. There may have been a word that I added in.
And also, par exemple, c'était pas pour le gilet, c'est parce que you said that because there are lots of things that I could work on. The art of thinking clearly, the paper that I have to read, and other stuff. There's a lot that I could do now, but I can't just like do 15 minutes of that. I cannot just decide, okay, now we're gonna start thinking about that. And in maybe 30 minutes, Clémence won't want to drive anymore, and we're just gonna put that away and do driving. Can't do it. Can't do it.
Testing. Okay, I think I just learned how to use my AirPods as a mic. I'm testing the dual capture feature, as well as the AirPod microphones.
Understand it. So I'm hoping you understand it more quickly. I'm hoping that my years of understanding it will help me explain it, or my years of trying to understand it. But you do not have a normal partner. It's kind of the reason you like your partner. It will never be like a thing where, oh hey Eris, I know that you're working on your essay today. Can you just real quick go outside and mow the lawn, and then you can switch back to your essay later. What? No. No, 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 no. Sometimes I'll be able to do that. Depends on what how into the essay. Because maybe while I'm mowing the lawn, I can be thinking about the essay. So sometimes I can do it. But we're not going to argue about it. If you say, hey, can you just do this? Oh, hey, Eris, remember, you got to do your work. Now, Eris, come on, just, do your thing now. Whoa, 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 whoa. Maybe I'll say, okay, I'm going to try. All right, let's get some music on. I don't know, blah, blah. I'm going to go smoke a joint, have a coffee, have a drink, whatever. I'm going to try, blah, blah. And then if I tell you, you know what? No, don't think I'm going to be able to do it now. We're not going to argue.